All right, guys, we're picking up today in our notes with translation. So, this is on the heels of our last day in class together when we lectured over the first part of the central dogma, right? So, we first did transcription and we talked about kind of making a copy of that DNA code. And now we're going to see where that mRNA copy goes and how it is read and a protein is actually created. So we'll start again with the location, goal, and then we'll get into the details. So for the location, translation is going to be happening somewhere in the cytoplasm on a ribosome. Now remember, we have attached ribosomes attached to the rough ER like we see over here, and then we also have free-floating ribosomes like we see out here. So the attached ribosomes are going to make proteins that are actually going to attempt to leave the cell. Some of them do. Some of them get embedded in the cell membrane on their way out, which is how we end up with proteins in our cell membrane. The free-floating ribosomes are going to make proteins on them that will stay inside the cell and serve the cell. But the location for translation, everything we're getting ready to talk about, is on or at the ribosome. Depending on where the ribosome is, the protein created on that ribosome gets a specific final destination. Now, the big goal of translation is essentially to read that messenger RNA that we just created in the previous process of transcription. So we want to read the mRNA and we want to use that as sort of a code or a template to be able to create a protein. Now, keep in mind, a protein is really just a string of amino acids. So think back to our beginning units on biochemistry, right? Proteins are one of our macromolecules, and proteins are made up of monomers or little subunits called amino acids. So ultimately at the ribosome, we're going to see, and here's a picture of the ribosome with its large and small subunit. Here's the mRNA kind of running through it. We're going to see some molecules right here help us put amino acids together and build a growing protein chain. So each one of these little dots coming out here is an amino acid, and the full product will be a protein. For the details, okay, so we see the same image again. For the details, we're going to take it in the same order. Big picture, we have initiation. In initiation, the messenger RNA is going to land on or encounter the ribosome. In elongation, we see that the ribosome reads that mRNA and starts to facilitate the process of binding together amino acids to make a protein. There is a starting sequence that is involved. The ribosome is looking for a starting sequence on the mRNA. So that, again, is very similar to what we have seen in transcription and even in DNA replication. Termination is going to be stopping the process, right? So there's going to be a stop sequence on the mRNA. And when that stop sequence is encountered, the mRNA and the ribosome sort of separate from each other and go their separate ways. In looking at the specifics of each one of these, so we just wrote down the big picture. Now let's talk specifics. In initiation, when we say that the mRNA lands on the ribosome, really remember the ribosome is made of two subunits. So here's the large subunit, here's the small subunit. They're going to kind of open up and clamp onto the messenger RNA that is free floating out in the cytoplasm, right? So mRNA was made in the nucleus, but then it escaped the nucleus through those little nuclear pores. And now it's free floating in the cytoplasm. So if this is going to land and encounter a free floating ribosome, they can kind of bump into each other. If it's going to an attached ribosome, the mRNA is going to have to move itself over to the rough endoplasmic reticulum. But one way or the other, the two encounter themselves. The large subunit and the small subunit sort of clamp down on the mRNA. And then this ribosome complex starts scanning the mRNA for what is called a start sequence. The start sequence on the mRNA is always AUG. And we call this the start codon because remember, we read mRNA in segments of 3 base pairs. And those 3 together are called a codon. So the starting sequence, the starting codon is always AUG. And so the ribosome will scan until it finds the start sequence, and then we can cue elongation. So here comes the next step of elongation. So we have identified our starting sequence, and the ribosome will start to sort of read the mRNA every 3 base pairs in sections, in codons. And so AUG is the first codon, and then the next 3 base pairs after, or the next 3 nucleotides after, would be the next codon. And then it would move down to the next 3 and the next 3, and it reads codon for codon for codon. As the ribosome is reading the codon, these little tRNA molecules, so let's look at this top diagram for a moment. These tRNA molecules are going to bring in appropriate amino acids based on the codon that was read on the mRNA strand. And so in floats the tRNA holding its little amino acid, and it will match up, in a sense, it will match up with the codon sequence that's being read by the ribosome. That's sort of like a checking mechanism to make sure that the tRNA brought in the correct amino acid. If it brought in the correct amino acid, it will release that amino acid to this growing chain that's growing out of the top of the ribosome. And so the checking mechanism that I just kind of talked about is matching the anticodon on the tRNA, that's the letters down here at the bottom of the tRNA, matching those against the actual codon sequence that's here on the mRNA. So base pairing rules apply, right? So let's take one of these, for example. Let's read this codon right here. So the codon reads AAA. The tRNA floats in, and as long as, think about what does A match with, what does A pair with? There's no T's in RNA, remember? So A pairs with U. So as long as the tRNA that came in sort of matches according to base pairing rules, so AAA would read UUU on the tRNA, as long as those two sort of pair up, then this tRNA knows it can release its amino acid onto this growing protein chain. So it's a little checking mechanism to make sure that tRNA brought in the correct amino acid. Down here is the same diagram. It's just a little more cartoony. So you have the ribosome right here, and you have the mRNA that's being read in the ribosome, and it's read every 3 pairs, every 3 bases, a codon at a time. And here comes the tRNA floating in with its amino acid attached. If the code matches, the bottom of the tRNA base pairs with the mRNA, that amino acid will be given off to and covalently bonded to the growing protein chain out of the top of the ribosome. And so we continue to grow this protein chain until we are cued, the ribosome is cued to stop, right? So let's talk termination for a second. The ribosome eventually will encounter what is called a stop codon. So a codon that codes for we're done. And when that happens, there's this little thing called a release factor that comes in that stops the whole process. And you can see here's the growing protein chain. Once we encounter the stop codon, the protein chain detaches and sort of becomes a free-floating protein, and the ribosome opens back up and ejects the mRNA out, and the process is done. So there are several types of stop codons, and we'll look at those in just a second. There's not just one, like there was one start codon, there are multiple stop codons. Once the stop codon is encountered, the protein is released, and it's going to have to fold up. So this is a throwback to our biochem unit again. Protein folding, remember this chart? We have primary, secondary, tertiary, quaternary levels of proteins. What is ejected out of the ribosome is the primary structure, the string of amino acids right here. That's not functional. So we're going to have to fold it at least to this tertiary level before it actually becomes functional. So there's some folding that's going to happen really quickly. And then the protein will be transported to wherever it's supposed to go, either inside the cell or outside of the cell. This diagram up here shows the mRNA is released, and actually that mRNA is going to be degraded eventually by the enzymes and the organelles that exist within the cell. So it kind of gets broken back down and chopped into little individual pieces, and those pieces, those nucleotide pieces, can be used again later. Let's watch a quick video of how this process occurs. So if you are used to pausing and skipping through, don't. Watch this process because it walks us through all three steps: initiation, elongation, and termination. It's a little creepy music in the beginning, but hang with it.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into three stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
The video did a funny thing here. So I'm going to back it up. You don't have to know the names of the sites that exist within the ribosome anymore, but the video does show you the A, P, and E site. And that's just sort of lining up the tRNA so that they can add the amino acids into the growing protein chain. Let me come back to right here real quick. Don't know what happened there, but...
And the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So that protein would have to fold up into a three-dimensional shape before it could become functional and transported. Okay, so here's the last thing I need for you. Remember in class, I gave you a whole bunch of papers. Well, we're getting ready to get out 2 of those papers. One of them, oh, I said purple. Actually, I think it's blue. One of them is a double-sided paper that has this funny looking chart on it. And if you sort of flip front back, you'll see there's actually a couple of versions of this chart. So find the blue paper for just a second. Pause the video and find the blue paper. And once you do, come back to me and let's talk about reading it. So this is an amino acid chart or a codon chart. And so basically this shows you the codons that are associated with all of the possible amino acids. So when we read our mRNA sequence, we know that the mRNA codes for a particular codon. This is how we figure it out. And so your paper, your blue paper, you have one sort of square chart on the front that kind of looks like what is big on the screen, but flip it over for a second and you'll see 2 more charts on the back. Those are the exact same. So the codons will code for the exact same amino acids on every single chart because this is a universal codon chart. The format of it can shift a little bit. And so I've given us 3 basic formats so that we could get comfortable using all 3 and being able to kind of understand how to read all 3. But every single time, the AUG codon on any one of these charts is going to code for the exact same amino acid. The AAU codon on any one of these charts is going to code for the exact same amino acid. So let's talk about how do you read this chart. So let's use the one that's on the front for a second. So we know that codons come in 3 letters. And so to find this, you would first, let's say we're gonna code out, right? So let's do a quick check. We're going to code out the AUG codon, the start codon. So I would first, my first letter is A. I'm going to use this axis to find the A. Then my next letter in AUG is U. And so that's the second position up here. So if I draw the A and the U columns together, I get this box. And so every codon in this box starts with AU. And then I can either look around in this box or I can use this axis over here to find the last letter and run it across. So what amino acid codes for the AUG codon? MET is the name of the amino acid. And so if you were asked, it really stands for methionine, but if you were asked to write the amino acid that goes with the AUG codon, you would write MET, MET. Okay, so that's a quick example. Let's try another one. So let's use the chart to look for a second. There's only one start codon. How many stop codons are there? So scan the chart and see if you can tell me how many stop codons there are. Did you get 3? There are 3 stop codons. Here they are: UAA, UAG, and UGA. They all code for stop, which triggers the release factor, and the protein is released and the process is over. Okay, so here we go. You need to practice this before I turn you loose on the actual assignment that you're getting ready to do. So if the DNA, I'm going to give you a sequence of DNA. If this is your DNA, TTA, CCG, GGA, I want you to first transcribe it. So what would the mRNA sequence be? And then I want you to translate it and tell me what the sequence of amino acids would be. So pause this video and do this check. I promise it will help you to know if you're doing it correctly before I just launch you out into the assignment. So pause the video, and when you're ready, come back and we'll check the answers. Excuse me. All right, so if you're back, then you should have a sequence of mRNA, and then you should have read the mRNA in codons over here on the chart and given me an amino acid sequence as well. So here comes the answer. Check yourself. The mRNA strand that complements, so if I am transcribing, which is what we did the other day in class, the DNA sequence, it reads AAU, GGC, CCU. And then if I translate that sequence, it means I read it over here on the chart. My 3 amino acids are ASN, that's this codon's amino acid, GLY, that's this codon's amino acid, and PRO, which is this codon's amino acid. So that is the skill of translating on the heels, actually, of transcribing. So you did them both together. All right, so your practice is a little bit more of this. You'll need your blue codon chart paper, and then you will need the practice sheet that looks like this. So find this paper. It's one of the ones that I handed you in class, and you're going to read and answer a few questions right here. And that's pretty straightforward. On the back, so once you find it, and if you don't have it yet, pause this video and go get it so that you can actually look at it with me, okay? So once you're onto the back, you have several different DNA sequences that you are asked to transcribe, which means give me the mRNA to the DNA, and then translate, which is use your chart and tell me what amino acids. Here's an example up here. So if this is the DNA sequence, here's the transcribed sequence of mRNA, and then here's the translated. Notice the full mRNA didn't have to be translated because what does the ribosome look for? A start sequence. So the protein started actually 3 codons in at AUG because that's our start sequence, and it runs until we encounter a stop. So don't do more work than you have to when it comes to reading your mRNA on the codon chart. Start at a start sequence and only code through a stop sequence, and then you can stop. All right, so this is something that you need to do and have completed by the time you come back to me in class. Okay, good luck. Email if you have questions.